Hello.
Hello, good morning, sir.
Good morning, sir.
सर प्रणाम।
नमस्ते नमस्ते।
सर महाराणा प्रताप पीजी कॉलेज जंगल भूषण गोरखपुर आपका हार्दिक स्वागत करता है सर आज के व्याख्यान के लिए।
थैंक यू
सर आप हमेशा महाविद्यालय में अपना जब भी आपको हम लोग आग्रह करते हैं सर आप अपना सहयोग हम लोग को निश्चित देते रहते हैं सर। आप एक प्रखर अपने विषय के विद्वान के साथ साथ सर आप एक सिद्ध पर्यावरणविद भी है सर। आप एक समाज सेवा के कार्य में लगातार सर लगे हुए हैं। तब मैं सर बिना किसी औपचारिकता के सीधे आपसे सर सादर निवेदन करता हूं कि आज का जो आपका सर व्याख्यान है स्टेम सेल, सर आप उस व्याख्यान को प्रारंभ करने की कृपा करें सर।
स्क्रीन शेयर कर दीजिएगा। I'm greatly thankful to Principal Dr. Pradeep Rao, MPPG College Jangal Bhushan, Head Department of Zoology Dr. Raghuveer Singh, Dr. Vinay Kumar, and Dr. Shiv Kumar, who has given me opportunity to interact with the postgraduate students of your college, particularly the Zoology Department. So let us start the topic. Stem cell, a novel biological tool in medical, food, environmental science. In the last two lectures, we have discussed cell signaling and diversification and differentiation and diversification of cells in embryonic stage, cell memory, positional values. Today we will cover the topic maintenance of differentiated cells in the body, and the prime concern with the maintenance is the stem cell. I'm presenting a slide video let us see how animal development proceeds. After fertilization, there is formation of zygote and there is rapid proliferation of embryonic cell. That embryonic cell differentiate, as I have already told in last lectures, in different types of cells that make up the adult organism. And when a whole organism is formed, what happens? There is a natural death process that is called programmed cell death. I will take this topic in next lecture, and the cell death may be possible due to some traumatic events or any accident. But when there is a programmed cell death in natural process, there must be some device through which the cell death is balanced, and that is by cell renewal. and that cell renewal is maintained by the, mostly by in the body is by the stem cell. Likewise, the programmed cell death, when there is abnormalities in programmed cell death, there is possibility of various diseases in the body. Cancer, autoimmune disease, neurodegenerative disorders, Parkinson, Alzheimer's disease. So in this way, in the body, when it is differentiated and diversified and form a whole organism, there is an activity of both cell death and cell renewal, and that must be balanced to maintain the working of the whole organism. So this is the cell death and renewal are the areas of forefront of biology nowadays in 21st century. So let us start renewal of cells. When a whole organism body is formed, so you can classify the cells into three forms: tissue with permanent cell, renewal by simple duplication, renewal by stem cell. Tissue with permanent cell, suppose they are, we will tackle one by one. Tissue with permanent cells, not all the cells in the body subjected to turnover. There are some cells, tissues in the body, which is generated in appropriate numbers in the embryos, and it is retained throughout the life. And that cells are nerve cells, muscle cells of the heart, auditory hair cells of the ear, lens cells of the eye. These are the permanent cells, and they are formed in the embryonic stage, whatever the number required in the body, and they are maintained throughout the lifespan of the. So there is no turnover of these cells, and if there is turnover, there is very small quantity in min. There are certain cells where proliferation of endothelial cells is required, and these cells proliferate by simple duplication process. For example, if there is any cut in the body, and in that area there is no supply, continuous supply of blood, and tissue deprived of oxygen, that portion of cut area, then they stimulate vascular endothelial growth factor. Now this vascular endothelial growth factor activates the neighboring blood vessels to form a capillaries in that area to make supply of the healing wound. And how this happens, it is only the simple duplication of the endothelial cell, which here you can see, this is the blood vessel, and here this endothelial cell prolongs towards the area where there is cut, there is less oxygen in the tissue, and there is simple duplication, and afterward there is formation of vacuoles, and these vacuoles form in the continuous cells, and thereafter they form a lumen in this area. And now this lumen proliferate into the region, and next when arterioles or venules and make contact with the supply of the blood in that particular area. So this is called angiogenesis, and this is done by the process of only simple duplication. Likewise, if you see the regeneration in liver cells, here also simple duplication phase is it is maintained. In normal course, liver cells are in G0 stage, and if there is any cut in liver, suppose if you cut the 2/3 of the liver portion, then hepatocyte growth factors are originated, and that hepatocyte growth factors initiate a duplication process of that cell, simple duplication process. There is proliferations of cells, and that liver within 14-15 days maintains the whole site. So in this way, if you have seen in blood capillaries and liver cells, the maintenance of cell is done by the simple duplication process. Whereas in case of you have seen in nerve cell, muscle cells, auditory hair cells, and lens cells, there is no need of turnover of and the number of cells are fixed. But the most of the cells in the body is maintained by the stem cells. And as you know, there is 220 types of cells in the body, but the number of cells exceeds the number of tissues found in the body. The number of stem cells are about 268. It means certain stem cells can make another type of other cells. What is the stem cell? The first thing is, what is the stem cell? The stem cells are the building blocks of our body. Every single cell in the body stems from this type of cell. Stem cells are undifferentiated blank cells that do not yet have a specific function. It is not terminally differentiated, that it is not at the end of the pathway of differentiation. It can divide without limit, and when it divides, always it forms a daughter cell and embark a course leading irreversibly terminally differentiated cells. So here you can see this is the stem cell, and when it divides, it forms a stem cell and another cell which then proliferate and differentiate according to the nature of stem cell. So that there is a continuous division of stem cell in different parts of the body to renew the death, to take place of the death cells which died due to the programmed cell. So now the question arises, what decides whether a stem cell divides or stays? What decides whether a given daughter cell remains a stem cell or differentiates? What way the differentiation of daughter cell is regulated after it has become committed to differentiate? ये प्रश्न उठते हैं कि कौन सी चीजें हैं जो स्टेम सेल को डिवाइड करती हैं. अगर स्टेम सेल डिवाइड हो गया तो वो एक डॉटर सेल बनाएगा और एक स्टेम सेल बनाएगा. तो कौन से कारक और कैसे ये डिफरेंशिएट हो करके डॉटर सेल से रेगुलेट होता है इनका डिफरेंशिएटेड सेल्स बनाने में. So these questions arises. So now the answer is that there are two possibilities: asymmetric division and independent choice division. In asymmetric division, as I have already told you, in embryonic stage, there are certain local determinant which works in differentiation process of the and differentiation and diversification of cells in the body. Here, when the cell fertilized egg divides, this determinant is here and it divides into two forms. So when cells containing this determinant portion becomes stem cell, and cells without determinants, it will terminally differentiate into the cells to renew that particular organ. So this is called asymmetric division. Whereas independent choice, this is called choice determined by the asymmetry in dividing stem cell. There is a choice determined stochastically and or by the environment. So here what happens, this stem cell divides in two, and the environment surrounding it, this stem cell, decide its differentiation, terminally differentiation. What happens in first division? It may be both cells become stem cell, or one cell become stem cell, one differentiated cell, or both cells become differentiated cell. In second division, this two stalk may form 4 stem cell or 3 stem cell, 1 differentiated cell, 2 stem cell, 2 differentiated cell, 1 stem cell, 3 differentiated cell, or all are differentiated cells. Likewise, here this is 2 stem cell, 1 stem cell, 2, are all 3 differentiated cells. It all depends upon their conversion depends upon the environment surrounding that portion. So this is independent choice, and these are the possible outcomes of the stem cells. So in this way, the environment surrounding the stem cell and situation, which that cell faces, is important for its determination into the differentiated cell or a stem cell. The first stem cell was studied in 1961 in bone marrow, and that by McLeod and James Still in 1961. And in bone marrow, they observed that there are proliferated stem cells, and these stem cells may be converted into myeloid stem cells or maybe lymphoid stem cells. That myeloid stem cells after 3-4 division forms colony forming cells mix, and this colony forming cells contains CFC, eosin, eosinophils, basophils, granulocytes, megakaryocytes, and bust-forming erythrocyte cells. So these forms eosinophils, basophils, and these granulocytes forms neutrophils, monocytes, osteoblasts, and macrophages, and megagranulocytes forms platelets. And bust-forming cells, erythrocytes, form colony-forming erythrocyte cells, and erythrocytes are formed. Likewise, here the lymphoid stem cells form colony-forming T cells and colony-forming beta cells in the thymus, and they form T cells and beta cells, and they are also stimulate themselves to form more cells to act as in the immune system. So in this way, this stem cells was first 1961 was discovered by McLeod and Till, and this erythrocyte bust forming cells is stimulated by the interleukins, and after that they form several divisions. They form colony forming cells and erythrocytes, and they are stimulated by erythropoietin and by several divisions about from a single pluripotent cells 2 to the power 11 mature erythrocytes are formed. So in this way, the number of erythrocytes lost each day is maintained by the formation of erythrocytes in the bone marrow. So it is very difficult to distinguish these all types of cells in bone marrow. They are embedded into the fibroblast along with the fibroblast cells in the mesh. And this is the first reporting of the stem cells. Likewise, stem cells also renew the gut lining. The intestinal wall inside the wall are always in contact with the harsh environment, whatever you are eating. And epithelial cells are removed continuously from the intestinal walls, and that epithelial cells continuous supply is maintained by the slow cycles 24 hours dividing cells of stem cell. It is here seen by the red. It is found inside the villi in the base, and now they divide, and after division they form rapidly dividing cell cycles 11 hours, and there were non-dividing differentiated cells are formed. So in this way, the epithelial cell of here you can see the epithelial cells are removed from the intestinal wall. When you are eating, digestion process is going on, cell is shedding, and now this is maintained by the stem cell. Here the stem cell divides to form the stem cell and differentiated cells. Now this transit amplifying cells 2 to 3 layers, and after that it forms absorptive epithelial cell as well as goblet and endoendocrine cells. So in this way, the stem cell maintains the epithelial intestinal wall structure continuously by making several cells in place of removed epithelial cells. Likewise, this is in case of intestine, where in case of skin, your skin is also very in touch with the harsh environment, and there is also removal of the epithelial cell from the skin upper top of the skin. So here what happens at the base in the dermis, there is a basal lamella, and above the basal lamella, this is the stem cell which is going to divide, and after division, that cell, prickle cell layer comes into the prickle cell layer, it's moved toward the prickle cell layer, and thereafter granular cell layer, it's shifted to the granular cell, granules are formed, and keratinized squamous layer becomes keratinized, and upper portion, keratinized squamous portion is formed, and here the squamous flake out from the surface. यहाँ से ये cells बाहर निकलती रहती हैं continuous, और वो इसके अंदर से stem cells के मेंटेन होती रहती हैं. ये लाइफ लॉन्ग चलता है, और इसीलिए आपकी स्किन हटती रहती है 15 दिन 20 दिन बाद, और रिमूवल डेथ होगा, और उसके बाद से नीचे से वो रिप्लेस होती रहती है. तो इस तरह स्टेम सेल इसको मेंटेन करता है. ये आप स्किन का स्टेम सेल का ये भी दो देखिए, स्टेम सेल है और ये इनर पोर्शन में पाया जा रहा है, इसके डिवीजन से ये डिफरेंशिएट होकर के ऊपर से निकलती रहती है एपिडर्मिस. एक one thing is that a hair follicle, in the base of the hair follicle there is bulge cells. These are also stem cells, and they form the hair. But in case of if there is any accident and there is this removal of this stem cell is removed here, suppose, so this stem cell, bulge stem cell of the hair follicle forms the another stem cell of the skin, and they can start this process. So there is interconversion from stem cells to this skin stem cells, and the number of stem cells, about 10% population of the basal portion of the skin is stem cell. There is, and that is continuously dividing and forming the skin in natural condition. So that it is called that the stem cells are immortal cells. It is from the embryonic stage when it is identified as a stem cell, it continuously divides throughout the life. Suppose this is a stem cell, red cell, it divides into another cell, differentiated. This stem cell is maintained. Again, it divides another cell. Now it is maintained. Now again, it divides from another cell. This is maintained. Will divide and again divide. In this way, this continuous division is going on, and they're differentiating in different forms in different organs according to their location. So that it is called, it is an immortal cell, immortal stem cell dividing throughout the life. Likewise, skin epithelial cells, there is a satellite stem cells in the muscles. And these satellite stem cells are very important as it controls the is if there is any injury in the muscle, to make the fill up that portion and continuously. And normally you can divide the stem cell hierarchy into this totipotent, pluripotent, multipotent, oligopotent, tribipotent, and nullipotent. When it is terminally differentiated, it is called nullipotent. And when it is in zygote, about 32 cell stage, that is called totipotent. It means if you separate each cells, you can develop a whole organism. As soon as development proceeds in embryonic stage from blastula to gastrula, these three germinal layer, they become specified, and their potency from totipotent to decreases and at the end, when terminally it is fully differentiated, it is nullipotent. So this is the hierarchy of the stem cells in the body, and this is different stem cells, what they maintain in the body, number of cell types, what they are making example of this. So what is the application of this stem cell? Till we have discussed how they maintain the body structure, such as skin, intestinal structures, and so on. All the portions of the body is maintained by the stem cell. So what is the medical application of the adult stem cells? So you have heard the bone marrow transplantation process when a person is suffering from a cancer. Suppose this is a person having a tumorous cells cancer here. So what is the therapy? That is, you have heard the name chemotherapy. High dosage of chemotherapy is given to eradicate this tumorous cancer cells from the body. And in due course, what happens when high dose chemotherapy is given, along with these tumorous cells, other body cells are also damaged by that chemotherapy. And the hemopoietic system, bone marrow hemopoietic system, is also disturbed. So what is the person who is going to take high dose of chemotherapy for the treatment of tumor, so there must be, if it is healthy in the bone marrow, hemopoietic cells are taken out from its body or its near relatives taken, and after chemotherapy, bone marrow transplantation is given to this person, and then that bone marrow transplantation restores the hemopoietic system of that particular patient. So that in cancer treatment, and this is one of the very good methods to restore the tissues after chemotherapy. And embryonic stem cell and therapeutic cloning is another aspect of medical application of stem cell. What is this? In embryonic stage, as I have already told you, in early embryonic stage, all the cells are totipotent. So that in embryonic stage, if inner cell mass is taken into the culture media, and now it is maintained in the culture, we can use these cells to form any cells of the body. How? Here, in the culture media, the embryonic stem cell is taken before early embryonic stage, and they are undifferentiated. Here, the leukemia inhibiting factor is included. If we remove the leukemia inhibiting factor, after removal of the leukemia inhibiting factor from this culture, these cells develop in embryoid bodies, and thereafter they form blood cells, epithelial cell, adipose, smooth muscle, nerve cell, hard muscle. Whatsoever the body cells can be formed from this embryonic stem. They are because they have capacity to form the whole cells in the body. They are totipotent. So with this help of technology, we will treat many problems in the health. You can see the first cloning by somatic cell nuclear transfer is done in 1997 by an dollies is the byproduct of this somatic nuclear transfer. In 1997, what they have, Wilmer done, he has taken unfertilized egg and removed the chromosome nucleus of this unfertilized egg. Thereafter, he take mammary gland somatic cell, adult, and they take the nucleus and transplant it into this egg, and it is transferred into this cell. And after transferring this cultured embryo is implanted in the foster mother, and then clone is formed in form of dolly. And this is the bony, which is a normal reproductive procedure produced by the normal reproductive procedure from the Dolly, the first sheep farm clone of the world in 1937, Wilmer. So this technology, stem cell technology, is cloning for somatic cell nuclear transfer, as well as it is also used in therapeutic cloning. In therapeutic cloning, if suppose there is a patient and it needs a transplantation of some nerve cells in the brain, so what happens in therapeutic cloning? The skin cells or adult somatic cells are taken from this patient, and thereafter unfertilized egg nucleus is removed from here, and from healthy cells of the patient, nucleus is taken off and it is transferred to this egg. This is cultured in the early embryo. Now from this embryonic cells are cultured in the petri dish, and now with the certain growth factors, addition of it differentiate into the nerve cells. Now these nerve cells can be used to transplant different area, whatever the damaged area. So this is stem cell technology is also used as a therapeutic cloning. And likewise, if there is a problem in genetic configuration of a patient, suppose this is a rat having a RAG2 deficient gene, and there is immune deficiency is found in this rat. So how it can be cured? So for this cure, what happens? Fibroblasts from this rat is taken and cultured, and this nucleus is fibroblast is taken out and transferred to the enucleated egg of the rat. And then this is cultured embryo, and then embryonic cells from this cultured embryo are taken into the culture media. Thereafter, a plasmid carrying normal RAG2 gene is transferred into this embryonic cells. And now this embryonic cells contain the RAG2 gene, which was not found in this rat. And now this differentiate into the homopoietic cells, stem cells, and transplanted into the same rat, this rat. And after transplantation, it restores the immune system deficiency problem, which was here, RAG deficient mouse. So in this way, you can correct the correction of inherited immunodeficiency by any disease. You can correct that diabetes, whatsoever the disease, it is genetically not genetically genetical deficiency can be cured by this immune system. This is the therapeutic uses of the, and one thing is very important, that stem cell technology now utilized to prepare meat in the lab to fulfill the appetite of the world. This is very recent research work. As you all of you know, that cattle farming is a big problem nowadays for the production of meat and milk, because the greenhouse gases emitted by the cattle farming is about 17.8%, about 18%, and it is higher than the transport section greenhouse gas emission. And you can see in developed countries, this population increase is not so problem, because at present the developed countries are 1 billion, 1.2 billion, whereas and that population is maintained in 2050. But in underdeveloped countries and developing countries, the population hike is very high, very fast, and up to 2050, there is a demand of billions of metric tons consumption, and so that if cattle farming is added by deforestation process, what happens? There is more emission of greenhouse gases, and that will cause harms to the environment. And you can see the red portion is the 70 pounds meat consumption all over the world. It is indicating the amount of pounds of the meat production and uses consumption. And you can imagine if you grow a 1 pound of potato, it means 0.17 mile running cars CO2 emission, 1 pound apple, zero point 0.2 miles, and pork and beef, it is more than 2.52 miles car running. So that you can imagine the greenhouse gases coming from this cattle farming. And you can see here, the cattle farming is for the livestock production is 18% next to the energy production, but it is more to the transportation section. So what happens? Now the scientists are making meat in the lab, and it may be possible in coming 4 or 5 years, there is a meat lab at every corner of the city, and you can go and take the meat, whatever the amount you need. And what is the technology? I discuss here. Suppose if you are going to be prepare a cow meat, so then or pig meat, whatever the animal, so you take an embryonic stem cell of this cow and then adult stem cell from its muscles and mix both into a culture media having a growth serum, and after mixing it will convert it into the embryonic stem cell. There is a fast division and adult stem cells coexistence make it into the stem conversion into the muscle cells. Now these muscle cells are scaffold on this net, and after exercise in medium, they form muscle fibers and they are peeled and they can be used for eating purposes. So whatsoever the meat you are taking, you can form chicken meat, fish, muscle, whatsoever according to the. And this work is done by the Netherlands by Professor Van Allen, Amsterdam University, and Professor Hacks, Utrecht University. The one thing is very important that the cost factor, they are working on this cost factor. And if the cost factor is going down, then this technology will be very beneficial. And due to this technology, what happens up to the meat demand vision 2050, scientists are confident that with the help of 10 stem cells, we can prepare 50,000 metric tons of meat in 2 months. So such a line would be sufficient to feed the world. A potential environment impact will be that the meat will be approximately 30 to 60% lower energy level will be involved in this technology. Greenhouse gas emission will be reduced by 80 to 90% by the cattle farm, and 90% lower land will be used, and that land can be used for the development of forest. So in this way, this meat production is solving, lab lean production will solve the problem of environmental impact. Like now the stem cell anti-aging and rejuvenation. How stem cells rejuvenate? Nobody wants to grow older. And stem cell technology is a fountain in which, with the help of this, by taking a bath in this fountain of the stem cell, you can become young for a long period. And so that in this technology, the cells to the embryonic stage is maintained. And this is a very, very new research area which can reduce the aging process and prolong the lifespan of the human. What is the characteristics of aging? You always see this process in your routine life. If you are young, your organs are robust, high tissue turnover. If there is any cut that is filled earlier, wound healing is rapidly, less inflammation, less scarring. And if you becomes older, the frail organs, low tissue turnover, wounds heal slowly, more inflammation and more scarring in the body. So how aging can be checked? How aging happens, first of all, we discuss. If we will discuss the role of stem cell in aging process, so you can say that blood cell, gut epithelial cell, and epidermal, which I have discussed earlier, they have high cell turnover, high regeneration, because these cells are rapidly lost in day-to-day life. The stem cell role is here more. When you come to the liver, skeletal muscle, adrenal cortex, it is lower than this portion, low turnover and high regeneration here. But when we move to the brain, kidney, and heart, there is low regeneration and low cell turnover of the stem with the help of stem cell. So in this way, the stem cell behave differently in different organs, and particularly brain, kidney, and heart, the turnover is less. So what happens? Suppose in case of stem cells in aging, if there is a less number of stem cell, decrease in number of stem cell in aging higher age group, or their capacity of the stem cell go down, or environment surrounding the stem cell contribute aging process. So there are three things. One is number of stem cell. Second is capacity of stem cell, or environment surrounding the stem cell contribute aging. Let us see. Here you can see there is a muscle cell, and in muscle, satellite cell, as I have told you, is a stem cell of muscle. So in young stage, if there is any damage in the muscle, the satellite cell activate and it renews to the satellite cell, as well as it forms a cells which differentiate into the muscle cells, and muscle fibers are generated. And this process is very fast. When you grow older, what happens? The satellite cell number is less, their capacity is less, and so that it is not activated in the process the young stage, and so that their differentiation is very slow, and this proliferation is slow, and this is generation of muscle is very slow. So, and this is due to this aging process, there is no such fast filling of the wound in as you grow older. Okay. What happens in muscle repair? This is a muscle fiber, and these are the green portions are satellite cells. If there is a wound is formed here, then satellite cell activates, it forms a differentiated cell and a satellite cell. And this differentiated cell forms a muscle fiber which fill up this space, or this differentiated muscle cells comes to the here, satellite cells, or it comes from the bone marrow portions, and they fill up this wound in normal course of muscle formation. In changing notch delta signaling, as I have told you, in cell signaling process, that this is very important in embryonic stage, as well as it is also very important to maintaining the muscles fibers in skeletal muscle in the embryo, as well as in proliferation and differentiation and declines in old muscle. So Notch delta signaling declines in the old muscle. Suppose if this is a satellite cell in the muscle and delta Notch signaling is going on, and what happens once a delta Notch signaling starts, this lower portion goes to the genes and activates the protein synthesis, and then actin myosin is formed to maintain this structure of muscle. But as soon as in younger muscles, the Notch delta signaling is more. Number of Notch delta signaling process is more, and they maintain the actin myosin after wound are in normal course. But when you grow older, this number reduces, Notch delta signaling, as well as satellite cells number is also less and their capacity also reduces. So that in old muscles, there is not so fast formation of actin myosin, and so that the wrinkles are formed on the body, and due to the less formations of myosin and actin with the satellite cells of muscles. To turnover is human heart a poor regenerator. If human heart muscle is visible, as I have already discussed earlier, skeletal muscle, human heart is a poor regenerator. Here the cell turnover is very low and regeneration is also, as I have already told it. And you can see here a heart which is formed in embryonic stage is just a minute, wait for just a minute. I take the mouse to start this video. Just a minute. Anyway, this video is not running. It is just demonstrating that the heart started contracting from the embryonic stage, and and it continuously contracts throughout the life and providing oxygenated blood to the whole body and taking unoxygenated blood from the lungs. And this cycle is going on throughout the life cycle. What is going on is that if there is a blockage in the heart, you have sent heard the word heart attack. What is the heart attack? Anyone? Heart is supplied by coronary arteries, and these coronary arteries, you can say, there is deposition of cholesterol. As the age goes after 20 years, this process is started in each and every person. And when our lifestyle and feeding habits are not good, there is deposition of this cholesterol. And by virtue of increasing, if there is any damage in epithelials and inner wall of the blood capillaries, due to the blood pressure or any other, there is deposition of blood clot at this point, and there is no supply of blood, oxygenated blood to this heart. And in this condition, you can see there is a blackening area in the heart, and this is the dead heart muscle due to lack of oxygen supply. So what happens in the heart attack? If there is a heart attack and there is no supply of oxygen because there is a blockage of this heart to reach this area, these cells become die, and these dead cells, like you have seen uh, a cycle tire in bulge area. If there is a weak portion in the tire, that area bulges outside, and cycling moving not in a straight way, it is jumping some way when you are running that cycle. Likewise, this heart, this becomes bulge area, and this heart is not able to pump the blood to the different parts of the body so efficiently, and so that there is a problem after that, after cardiac arrest. Here you can see the normal cells, and when there is lack of oxygen due to the non-supply of the blood to that particular area, they are necrotic cells and they become damaged. And what happens? Heart regeneration is lost in the evolution. If you goes from the fish to amphibia and mammal, as you proceed in evolution, the regenerative property of the heart decreases. In case of fish, it is fully regenerated. If you cut the fish uh, um, heart, that will regenerate immediately after some period. Whereas, because there is a regenerative property, but if you cut the heart of a man, it is not able to regenerate. So you can heart cell therapy by heart failure. A heart regenerate by tissue displacement in the fish, where in case of human, heart failure is caused by cardiac tissue loss. So can we replace heart cells to treat the disease? Can we stimulate the heart attack to make new cells? Can we keep new cells alive? There are probability how we can treat the heart failure or heart damaged damaged cardiac cells in the heart failure. So once a change of heart stem cell may transfer the way doctors speak. This is the biggest revolution in the medicine. With the help of stem cell, heart failure or cardiac dead cells can be replaced. And for the first time in 2009, Mike Jones, a patient which received an infusion of own cardiac stem cell in 2009. What happens? Dr. Robert Bawley of University of Pennsylvania meet Mike Jones in 2009, and Mike Jones is not able to move even a 5-10 feet with area, and so that he suggested that he will do a cardiac stem technology with the help of cardiac stem regeneration into the infusion into their heart, and he has done that infusion of the cardiac cells, and there is a 40% recovery in that fellow, and they becomes doing walking and doing some works started. And in this way, what Robert Bawley has done, he has taken stem cells from the cardiac cells of that fellow, Mike Jones, and make a culture of that stem cell, and then make infusion of that stem cells into the heart of that fellow. And he, after 14-15 days, he found that there is 40% recovery in the cardiac cells. Now, in normal course, there is 1% replaced, cells of the cardiac cells are replaced, about 4 to 5 billion of muscle cells each year by simple duplication process. It is a very slow process. But with the help of stem cells, if there is a cardiac failure and there is cardiac cells are not working, this stem cell technology can infusion of this stem cell technology can be very beneficial tool for treatment of cardiac arrest in coming years. And likewise, stem cell is also involved in forming organs. And super glue, and this is the super glue when the matrix cells are embedded into the super glue of the natural environment of the tissue. Now the scientists are using this fibroblast and super glue for formation of inner anatomy organs, जैसे kidney, and that kidney is first of all Anthony et al. Of the Wake Forest Institute Regeneration Medicine showed depositing the several layers of the nephron units with the help of 3D printing and inkjet printing, and instead of ink they use living cells, and thereafter they make a whole kidney of human kidney. This kidney is formed, but there is no working blood vessels and tubules for collecting urines. For that purposes, they uses a sweet solution to this problem with the help of super glue. They dissolvable sugar modes of vessels. They built first and then built up the appropriate cells around the mold. And later the mold is washed out and forming the passageway that is able to start up varying blood pressure levels found in the blood. So in this way, they are able to manufacture a large scale to forming a kidney and for transplantation process, and they are using liver transplantation, kidney transplantation, this technology. And likewise, for the treatment of Parkinson's disease, they have also used this technology, and for neurodegenerative disorders like Parkinson's, Alzheimer's, and with the help of embryonic stem cells, they use that to form the nerve cells, and in coming years, this stem cell technology can solve this problem. So that in this process, we are taking that embryonic stage, there must be a need of OR there, and so that there is some ethical problem in doing this stem cell technology. Treatments are studied. But in 2006 and 2011 is very crucial in this technology procedure, because induced pluripotent stem cells and parthenogenetic stem cells are produced, and they are the revolution in stem cell technology procedure. But more than 900 laboratories all over the world is working on this technology. And what is the induced pluripotent stem cell? You can prepare a stem cell from any cells of the body. In normal course, what happens after fertilization? Embryo grows into germ layers and thereafter differentiate into different types of cells, as I have told you in earlier lectures, and they form different types of cells terminate. Now, this technology is going on that with the help of these differentiated cells, we can make induced pluripotent cells by reprogramming. And this clock, biological clock, which is coming from embryonic stage to differentiation germ layer, differentiated layer, and terminally differentiated cells. Now, with the help of reprogramming, we produce induced pluripotent cells, and we can reverse the biological clock here. And by reversing this biological clock, several problems can be solved in biological science and medical science. And I'm not going so detailed. And this work, Sani Yamaka of the University of Toyota and his colleagues in 2006 found that only 4 genes, ARC4, SOX2, KL4, or CMYC, are necessary to produce induced pluripotent cells. And for this, Sani Yamaka got a Nobel Prize in 2013. And our Prime Minister, when visiting the Japan for the first time, meet this fellow, Nobel laureate Sani Yamaka, who has discovered that with the help of this gene, you can change any cell of the body into the pluripotent stem cell. And what he has done? He has taken on 4 genes in retrovirus and with added in retrovirus, and then it transforms it to the mouse skin cell, and then with the help of this, he produces induced pluripotent stem cells. Later on, another group, the Harvard group, Dr. Konar. They have reported that with the help of retrovirus, they are permanently attached with it, so that they have used adenoviruses and even non-integrating viruses, and with the help of they produces induced pluripotent steps. Later, the scientists have reprogrammed that even the proteins developed by these 4 genes are able to produce induced pluripotent stem cells. And thereafter, they have, with the help of fluorescent technique, they have identified the within 15 days, this with the help of this reprogramming, they have developed induced pluripotent cell and identified with the fluorescent technique. So that because it is very crucial that if you have to select the induced pluripotent cell colony, and because if failed reprogramming is used, then tumor may be developed. So this is for the very important to separate these cells precisely, and with the help of fluorescent technique, it is isolated. So this is a wonderful research which is now going to be a milestone in the field of stem cell technology, that you can produce any cell, any organ from any part of the body. And here, what as I have told, that OSKM factor is very important, and master gene regulator protein OSK induces both their own and other proteins. So this KLFA, it KIF OSKM, OSKM. So now they are initiating each other and self both in this procedure, or self-sustaining feedback loop, and maintain cells in embryonic stage. By this activation, they also activate permanently induced expression of nanogenes. And with the help of this, there is upregulation of embryonic stage genes and downregulation of differentiation genes. And MIC is upregulation of cell proliferation and loosening of chromatin structure in induced pluripotent stem cells. So this is the technology which is working. And here you can see if a fibroblast is studied to select cell to convert it into pluripotent cells. So gene FXB is present in all cells, but it normally expressed only in embryonic stem cells, this gene. And a fibroblast, genetically, this is the G4180 resistance gene, which is not able to make protein synthesis start. With the promotion of FBX gene activation, this portion is activated. Here, if there is no activation of this gene, then normal cell die. But when the activation of this one, what happens? The rare change instead iPS character resistance gene expressed, and induced pluripotent cells are survived. So this is the techniques which is working with the OSKM factors, which I have already told produced by the OSKM genes. And now how it converts the induced pluripotent cells active OSKM expression. The first step is transcription wave 1. So if suppose this fibroblast is going to be converted into the induced pluripotent stem cell within 15 days, so what happens? The first of all, when transcription wave 1 started, up to the 9 days, loss of fibroblast characters, hello,
जी सर आ रही है।
आ रही स्क्रीन नहीं जा रही, स्क्रीन शेयर गड़बड़ हो गया लगता है। हेलो,
यहाँ से तो ठीक है सर।
यहाँ कुछ प्रॉब्लम आ गई। Anyway, so these are the technology which is involved in formation of the stem cell technology used for different purposes. And the last one, which I'm not able to produce you because there's some problem in my computer, parthogenic way. Then California-based stem cell technology laboratory has developed a stem cell without any fertilization. You can start stem cell formation, and so that they called it parthogenic stem cells. And parthogenic stem cells is able to maintain the same character without fertilization, and there is no ethical problem in this. So in this way, we can use this technology for different purposes, for medical, health, environment, and all these purposes. And a stem cell which is involved in the maintenance of the whole body is very important, and its name is stem cell because stem is word is used in plants to holding the whole plant. Okay. Here, these stem cells are maintaining the whole body, so that they are called stem cells. Okay. So this is all about the stem cell. If you have any question, you can ask. Hello. Hello.
हाँ जी सर, आवाज आ रही है आपको?
And if any question, you can ask.
कोई विद्यार्थी प्रश्न पूछना चाहता हो तो सर से प्रश्न पूछ सकता है।
Hello.
Hello.
Sir, I have to ask a question. Sir, as you have discussed that liver cell can be regenerated, sir, but I want to know if the liver is fully damaged by some other cause or injury. So can we now develop new liver cell or new liver fully organ with the help of a stem cell?
And this technology is going on with the help of stem cells. In case of rat, it is not applied. In case of rat, it is totally transplanted that stem cell prepared livers in the rats. And this technology is now in a stage of research. And I think that within 4 or 5 years, we are able to transplant different organs of the body, not even liver, kidney, and many organs can be transplanted with the help of this technology.
Okay, sir. Sir, a question more. Sir, can we keep the liver cell alive of a cadaveric donor? Sir, transplantation of a cadaveric donor is also possible in liver. I think.
Categorically, whatsoever the question is, transplantation by donor, but that
Yes,
is
sir.
not. I think that is not so efficient, because there is different problems when a person is donating an organ to another person. There is immune system problem, there is acceptance problem of that organ. And if we are able to develop stem cell by the help of this technology, with the help of, suppose you have to transplant kidney or liver, that stem cell is produced by the same patient's skin cell, induced pluripotent stem cell, and with the help of that induced pluripotent stem cell, you can convert into the liver or kidney, whatsoever the organ you are going to make it. And with the help of that, because the genetic configuration of that organ will be same, and so that there is no chance of rejection in that type of stem cell developed organs to the same patient, because the genetic gene configuration is same. So that is, I think that 4 or 5 years, when this technology will be running properly, that will be a boon for the man.
Okay, sir, a question more. If we got a scar by an accident or an injury, uh, sir, satellite cells are capable of producing or regenerating same identical cell.
Okay, huh. We can develop with the help of satellite cells or the stem cells for the muscle. Muscles can be regenerated by the satellite cells, one thing. And above the muscle, there is epithelial cell. The stem cells in skin cells can develop the epithelial layer with the help of skin cells. And so that transplantation of filling of muscle cell and thereafter filling of the epithelial cell is important, and in transplantation procedure that can be done easily.
Okay, sir. Thank you, sir.
Thank you. Any other question? No, thank you.
बहुत बहुत धन्यवाद सर। स्टेम सेल जैसे विषय पर आपने जितनी सरलता और सहजता के साथ हम सभी का मार्गदर्शन किया, निश्चय ही ये हम लोगों के जीवन में बहुत काम आएगा सर। और ये इसी आशा के साथ कि जब भी महाविद्यालय परिवार सर आपको याद करता है, आप सरलता के साथ उपलब्ध हो जाते हैं सर। अतः मैं महाराणा प्रताप स्नातकोत्तर महाविद्यालय जंगल भूषण गोरखपुर की तरफ से आपका हार्दिक आभार व्यक्त करता हूं सर। और साथ में हमारे व्याख्यान में डॉक्टर प्रदीप सर भी जुड़े हुए हैं, मैं उनका भी महाविद्यालय परिवार की तरफ से हार्दिक आभार व्यक्त करता हूं सर। प्रणाम।
थैंक यू।